So when we talk about enzymes, what do we mean by that? What are enzymes? Are enzymes lipid? Are enzymes carbohydrate? Are they nucleic acid or protein?
Proteins.
Proteins.
So
Catalyst
yeah.
of certain reactions po.
All right. So when we talk about the enzymes, these are special set of catalysts. Actually, they are proteins. So they came from the Greek word enzymo, meaning to say they're inside the yeast. So what does the enzyme do? They accelerate the reaction rate within the cell so that the reaction rate would be compatible with life. So can you just imagine if you don't have enzyme in the body? So it's so impossible for life to be quite fast without enzymes. So for this lecture, we'll be tackling the part 1 and part 2. Okay, so we'll be finishing the entire lecture of enzymes. So this would be my lecture outline. So we'll be discussing the enzyme characteristics and mechanism of actions. Okay naman ako, no? Malakas naman yung boses ko.
Yes, Doc.
Yes po.
We'll discuss enzyme structure, nomenclature, factors affecting the enzyme. We'll discuss the enzyme regulation and classification of the enzyme, and then we'll go through with the enzyme kinetics and inhibition. Okay, so part 1. So as I mentioned a while ago, you have to know before the end of this lecture, I want you to be able to define and name the enzymes. You have to discuss the properties and structure of the enzymes, discuss and explain the mechanism of enzyme catalysis, and discuss and explain the enzyme activity control. So as I mentioned a while ago, life wouldn't be possible without enzyme. Now, when there is a deficiency in the enzyme, it would lead to diseases. Okay? So aside from that, they're using enzymes right now because they can therapeutically target some problems that make enzymes as your valuable drugs. Okay? So one that drug that is used as an enzyme, okay, would be your amylase. Okay, when a patient has pancreatic problem, they're given an enzyme or a drug that is called an amylase. So when we discuss later digestion and absorption, you learn the importance of amylase. So binibigay nila yon to help the patient in digesting some of the carbohydrates that the patient takes in. So aside from that, you'll see in your SGDs, in your conferences, enzymes are also used for diagnostic markers, no? Like for the different types of your cardiac problem, the different types of pancreatic problem, we're making use of these enzymes also to check if there is damage in the muscle or damage in a particular tissue. Even in liver problems, okay, we're checking some alkaline phosphatase, okay, to check if how extensive the damage in the certain organ. So it is utilized as analytical reagents in research also. So again, what are enzymes? Enzymes are protein which accelerate chemical reactions. So some enzymes are ribosomes. Okay? So they regulate metabolic rates and their biological catalysts. Okay? So when we talk about the enzyme, so how do enzymes catalyze the reaction? So any reaction will be corresponding to substrate and reaction in an end product. Okay? So make sure before the end of this lecture, you'll be able to know what is a substrate, what is the reactant. Substrate and reactant, by the way, are just the same term. And what is the product? Okay. So when you have substrate, okay, undergoing a chemical reaction, you'll be producing a product. Okay? So for a reaction to be spontaneous, you'll be having the free energy of the product, which should be less than the free energy of the substrate. So spontaneous doesn't necessarily mean that the mechanical reaction or the chemical reaction would proceed at fast rate. Okay? The rate of the reaction kasi will depend on the transition state, which is the energy required for the transition state. So when we say transition state, what do we mean by that? This is the state between the substrate and the product that the substrate has to undergo before becoming a product. So that is your transition state. So for example, you were an infant and then right now you're studying to become a medical doctor someday. So the process of your infancy up until right now is what you call the transition state. And then later on you see the product. Will you become a doctor or will you quit? So the product would be dependent on the state between the substrate, the transition state that would happen. So the energy difference between the transition state and the substrate is what you call the energy of activation. So any reaction would have a corresponding energy of activation. The higher the energy activation, the greater the energy requirement for the reaction to proceed. Okay? So this is what I'm mentioning a while ago. So you have your energy of activation, as you can see here. So if you don't have a catalyst, if you don't have an enzyme, okay, you have a reactant there and then you have a product. Okay? So as you can see, the energy of activation is quite high. You have an increased energy of activation because you don't have an enzyme. As I mentioned a while ago, the higher is the energy of activation, the greater the energy requirement for the reaction to proceed. So, effort ka talaga, no? Unlike in a reaction where you have an enzyme, okay? So, the energy of activation is lower because you have an enzyme there that makes your reaction faster, proceed at the spontaneous rate. Okay. So again, when we say spontaneous, it doesn't mean we are proceeding at the fast rate. Okay. We are saying here that the energy of activation is lower. So the bottom line here, enzymes lowers the activation energy necessary to transform reactant into a product. Okay. So ganito lang yon. Kapag nag-aaral kang mabuti, that's your enzyme. Okay? If you are studying, if you are preparing very well in your exam, okay, in your SGPs, okay, you are actually making your medical school spontaneous. But if you don't study, you don't make your medical school spontaneous. So anong mangyayari? Babagsak kayo, dadagdag yung taon for your medical school. Tatagal siya lalo. Unlike if you study well, if you know the concepts, you know what you're supposed to know, you are actually decreasing the energy of activation. Thus, you're producing your products spontaneously. Okay? So enzyme catalyzed reaction pathway has a small energy barrier, no? Which is an activation energy to overcome. So in the presence of enzyme, the temporary formation of enzyme substrate eventually the formation of enzyme product lowers the energy of activation. So the enzymes catalyze reaction by lowering the energy of activation. The enzyme should bind to the substrate. Ito yun. So you have the enzyme, you have the substrate. When they bind together, you would produce what? You would produce your product. So the enzyme binding to the substrate, you have to remember the enzyme would have a unique shape that it allows it to fit to the substrate or to the reactant. So dapat swak siya. No? Kapag hindi kasi siya swak, magkakaroon tayo ng problem with that. No? So in the presence of enzyme, in an enzyme catalyzed reaction, the energy of activation is much lower, thus the energy barrier is easier to overcome. So paulit-ulit natin yan sinasabi ha. If you have the enzyme, you have greater or lower, you have rather lower energy for your activation of energy to happen. So you have lower energy there. So the reaction proceeds at a rate that requires lower energy, making it faster. So the enzyme has to bind to the substrate, and the binding site is known as what? The active site. So you see in this diagram, you have active site, you have the allosteric site, right? So the binding of your enzyme and your substrate should take place in the active site. Now, any other site on the enzyme other than the active enzyme is known as allosteric site. So, kung hindi siya magbabind doon sa active site, ang tawag na doon, allosteric site. Okay? So, in enzyme substrate binding, okay, we have different models. Okay. One model would be the lock and key model. So, lock and key model is a model wherein the enzyme's active site fits the substrate perfectly. Okay. So, ibig sabihin, soulmate mo siya, nakita pa lang kayo, alam niyo na sa isa't isa, para kayo sa isa't isa. Swak na swak kayo, personalities nyo, likes and dislikes nyo, hobbies nyo, swak na swak kayo lahat. Isang pag-uusap nyo pa lang, alam nyo para sa isa't isa na kayo. Yun ang tinatawag na lock and key model. So when the enzyme approaches the substrate, there is already an initial perfect fit between the substrate and the enzyme's active site. This explains why enzymes are very specific when it comes to substrate. So if this enzyme is acting on a protein, any other molecule that is not a protein, for example, carbohydrate like your glucose, like your galactose, would not fit to the active site. Therefore, would not be acted upon by the enzyme. Okay. So the problem with the lock and key model, the limitation is there's a limitation here. Because the limitation here is that it implies that your enzyme is very rigid. Diba? Ibig sabihin, para doon lang siya. Wala na, hindi siya, pagka iniwan na siya nitong feeling niya na perfect na tao para sa kanya, wala na. Wala na siyang mahanap na iba. Kasi nag-iisa lang yun para sa kanya. Okay? So parang sinasabi niya, isang enzyme, isang substrate lang. Okay? So, is this true for all enzymes? Kung true to for all enzymes, mahihirapan mag-take place lahat ng reactions natin. Because this is quite far true from a lot of enzymes because enzymes are protein and we all know proteins are quite flexible molecules. For this lock and key model, they're quite very rare. No? Very few enzymes would act like this one. So although they're very specific when it comes to substrate, they're not really specific. So as long as the substrate are interrelated to one another, the enzyme can still act upon it. So we call that, okay, so this is your lock and key. So we call that what? Induced fit. So si induced fit naman is initially hindi perfect yung active site mo. Hindi rin perfect si enzyme mo. So what happens here, when the enzyme approaches the substrate would induce a change in the active site. So it would complement the change or it would complement your enzyme. So they complement one another. So it's like, for example, first impression nyo sa isa't isa nung nagkakilala kayo, hindi kayo swak. Pero later on, habang nag-uusap pala kayo, okay pala kayo, ayos pala kayo, no? Nagiging okay pala kayo, you complement one another, you're bringing the best of one another, if not the worst of each other, no? So you complement one another. So that is what you call your induced fit model. So there is a modification of the lock and key model. So it implies the substrate can change the enzyme by inducing a conformational change in the active site. It also implies that enzyme can change the substrate by acting on it and turning it to a product. So this explains bakit yung mga enzymes would have a wide range of related substances. For example, proteases. So when we discuss digestion and absorption, you will see that there are a lot of proteases there that would act on a specific peptide bonds which are adjacent to a specific what? Amino acids. Okay? So that's how you're induced fit. So pinakamaraming type of enzymes is actually falling on the induced fit model. So now, A while ago when I discussed my outline, I also want you to learn at the end of the lecture the structure of the enzymes, the possible or the terms when we are actually defining the enzyme. So these are actually the different enzymes. We have the simple, the complex or hollow enzyme, under which would be your apoenzyme or cofactor, and under cofactor would be your prosthetic group or your coenzyme. So let's discuss them one by one. So when you say simple enzymes, they only made up of protein, nothing else. Okay? So when you say holoenzymes, so these are actually the entire enzyme together with the necessary cofactor plus the protein portion. So they're active enzyme which is with its non-protein component. Okay? So when the enzyme is without a non-protein moiety, you call that apoenzyme, and it is an inactive. Okay? So again, a holoenzyme is a complete enzyme. It has your active site, it has your cofactor, so it has your apoenzymes. Now, an apoenzyme, if you take out the cofactor or the coenzyme, it would leave the apoenzyme naman. So kapag walang active factor or active site, walang cofactor, you call that what? Apoenzyme. So apoenzyme is the protein portion of the holoenzyme minus the cofactor. Okay. Please remember the terms ha, kasi if you don't get the basic terms, if you don't know the basic terms of the enzymes, you won't be able to pass the exam. Okay? So, structures of your enzyme would have your apoenzyme plus your cofactor or your coenzyme. So when we talk about the cofactor, these are non-protein chemical compound that is bound either tightly or loosely to an enzyme and is required for catalysis. So we have two types of cofactors. We have the coenzyme and prosthetic. So when we say coenzyme, these are big non-protein component or organic molecules that are required of some enzymes to activate them. They participate in binding to the substrate. Okay? They bind to the active site to prepare the active site for substrate binding. So, actually, si coenzyme wala siyang catalytic activity. Ang ginagawa lang niya, tinutulungan lang niya para mag-proceed yung activity. So, they help catalyze reactions by donating or accepting electrons. They can transfer groups. They can form or break covalent bonds. And, of course, they can also provide functional groups. Your common coenzymes would be your lipoic acid. Okay? You would learn later on when we discuss your amino acid metabolism that lipoic acid is a factor that decarboxylates your alpha keto acid. So I don't know if it was discussed to you when you were discussing your amino acid, if alpha keto acid was touched. No? So an alpha keto acid is an amino acid wherein you take out your what? You take out your NH portion. No? So your NH or your CO portion, wherein you decarboxylate the carboxyl portion, leaving it to become your alpha keto acid. Another coenzyme would be your NAD or your NADP, your FAD. So what does it do? As I mentioned a while ago, the coenzymes can donate and accept electrons. Thus, your NAD, the NADP and FAD can undergo redox reaction. They can transfer electrons, they can undergo dehydrogenation, they can transfer hydrogen. So another coenzyme would be your CoAsh, which is actually involved in the Krebs cycle and involved in the beta oxidation when you discuss your lipid metabolism later on. Example that I mentioned here in the slide would be the vitamins. Okay? The body needs vitamins such as ascorbic acid. Do you remember what's the function of ascorbic acid? Do you remember?
Say cofactor.
Factor po.
Yeah, cofactor of what?
Collagen synthesis.
Collagen.
That's good. So you see that your ascorbic acid is one of the very vital factor for what? For the collagen synthesis. Okay? So aside from that, you'll be discussing later on in the amino acid metabolism the importance of your cyanocobalamin, which is your vitamin B12, your folic acid, which is your vitamin what? B9. No? So by themselves, they do not provide energy, but instead they help unlock energy by acting as coenzyme. What about your prosthetic groups? So these are again certain cofactors or coenzymes that are difficult to separate from the protein portion. Okay, so examples of which would be the heme. Okay? When you discuss your porphyrin synthesis, okay, you'll be discussing the heme. So you'll see there that the structure is not just hemoglobin, but it also contains different enzymes such as catalase. So when we talk about enzymes, malalaman mong enzyme siya kapag merong AST sa dulo. So, pag may AST siya, enzyme yun. So, for example, urease would act on your urea, right? Just like this one, okay, your DNA polymerase, okay, would be acting on your DNA, diba? Okay. So when you have the DNA polymerase as your enzyme, the reaction is called as what? DNA polymerization. Okay, polymerization. Okay. So you discussed that when you discuss your nucleic acid, right? So proteolytic enzyme, the suffix is, some proteolytic enzyme wouldn't have ACE as their suffix. So hindi lahat ng enzymes yung ACE. May enzyme kayo na ang suffix niya is nagtatapos sa -ine. Like your chymotrypsin, pepsin. Chymotrypsin is an enzyme responsible for um, hydrolyzing the peptide bonds, which are adjacent to your um, non-hydrophobic amino acid. Your pepsin is found in the stomach, which is actually secreted by your chief cells. Okay? And initially, they're non-active enzymes, which are your pepsinogen. They are activated by the hydrochloric acid that is secreted by your parietal cells, making them active to become pepsin. So pepsin is an enzyme also that hydrolyzes the peptide bonds when you eat something that is a protein rich. In additional, hindi lahat ng enzymes mo nagtatapos sa ACE because you have what you call the restriction endonuclease. These are your E. coli, your MST2. No? So they are involved in the bacteria. So can you name this enzyme? See the reaction. Pyruvate.
The carboxylase.
It's very good. So you see the pyruvate, you see the carbon dioxide. So that's your reaction there. So this is your enzyme pyruvate decarboxylase. So now let's discuss what are the factors affecting enzyme activity. So you have the different factors. So let's discuss them one by one. So for temperature, so for most mammalian enzyme, the enzyme best work at 37 degrees Celsius. Kaya nga yan yung normal, di ba? Yun yung normal na ano natin, na temperature natin. Please lang I'll take this call. So, as I mentioned a while ago, when we talk about the temperature, so most mammalian enzymes work best at 37 degrees Celsius. No? So sa atin kasi, yan yung normal temperature natin. So if we isolate the enzyme and lower down the temperature, what would happen? It would result to the inactivation of the enzyme. Pero the enzyme is not destroyed. It is still recovered when it slowly reached to the 37 degrees Celsius. Kaya, di ba may mga lumalabas na medical therapeutics na hina-hypothermia nila yung body, no? The rationale behind that is, to stop the reaction first, okay, while they're trying to fix what is wrong. And then, slowly putting back the patient into its normal temperature again. So, if we increase the enzyme naman, it would usually increase the activity of the enzyme to a certain point. Like for example, when you have fever, you would notice that your metabolic rate is quite increased. Kaya nga ang nangyayari sa inyo, nalalamigan kayo, di ba? Kapag may fever ka. Kasi you have an increased metabolic rate. So, you'll be needing what? An increased temperature for you to feel, no, na hindi ka malalamigan. So, pero actually, habang tumataas yung temperature mo, you'll feel colder. So, the thing is, there would be an increase in the enzyme activity when there is an increased temperature, but to a certain point. So, when they reach that certain point already, like for example, they reach 42, 43, usually you'll have seizures na, no? And then you can die because of that. So, the enzyme here, what would happen is that the enzyme would be lost or destroyed because of an increased temperature. And when the enzyme has been denatured, you cannot recover it anymore. So kaya ang mas nag-cling sila more on the therapeutics would be the hypothermia than the hyperthermia, right? Because in hyperthermia, you can destroy what? Enzymes. Environmental reaction. So the rate of reaction increases as substrate concentration increases. Okay? But again, to a certain point. So mas maraming substrate, mas mabilis yung rate of reaction. So for example, mas mataas ang grades mo pag mas marami kang pasado na exam. Mas mataas yung grade mo pagka pasado na yung exam mo, magaling ka pang mag-report, mataas pa yung major exams mo, siguradong papasa ka, mas mataas talaga yung grade mo. So it's the same thing. So if you increase the substrate concentration, the rate of reaction would increase. But then, for example, you got 100 sa shifting, so you got 100 sa major prelims, you got 100 sa SGP. Okay? Hindi na tataas yung grade mo sa 100. There's still a limit to that. It would come to a point that, even if you add a lot of substrate already, you won't increase your enzymatic activity. So it's the same thing. So maximum activity, of course, when the enzyme is saturated. So kasi kung ang grade lang, ang pinaka-grade mo lang is 100, 100 lang. So it's like this also, no? Sa mag-boyfriend or mag-girlfriend, no? Yung boyfriend, lagi niya niloloko yung girlfriend niya. Si girlfriend, pinapabayaan lang, no? So, si boyfriend, ini-increase pa rin yung substrate concentration. Niloloko pa rin siya, lahat pa rin ng kalokohan, ginagawa pa din niya. So, si girlfriend, okay lang din. Ini-increase naman niya yung patience niya, no? But then there would come to a point na ma-increase na yung maximum point ni girlfriend na wala na siyang patience, okay? Matatapos na din yung relationship because of that. So, that's your substrate concentration. There would be always a maximum activity. Kaya huwag niyong susubukan. Okay? So, enzyme regulation. So, you have your another, before I proceed to the enzyme regulation, another factor that affects your enzyme would be your pH. When you say pH, what do you mean by that? This is the power of hydrogen. Okay. So for most mammalian enzyme, the enzyme works best at what pH? At pH 7. Okay. So if we increase the pH, this would result to the inactivation of enzymes, making the enzyme denatured. Okay. However, in general rule, that's the general rule of the enzyme pH 7. However, when we discuss later digestion and absorption, you would learn there that few enzymes work best at lower pH. Like for example, enzymes found in your stomach, which is your pepsin. Pepsin works best at around pH 3 and below. Kasi remember, hydrochloric acid is around pH 1. So how come your pepsin, which is an enzyme, which I mentioned a while ago, works best at pH 7, can work best at pH 3 and below? That's an exemption. Your pepsin. Another enzymes would be your intestinal enzymes. Okay. Sabi ko kanina, 7. Pero if you increase the pH, you are actually destroying or denaturing the enzyme. But it is actually different case for your intestinal enzymes. Okay. In intestinal enzymes, okay, because the food coming from the stomach is acidic in nature, hindi naman siya po pwedeng pagpunta sa intestine, acidic pa rin siya in nature. Bakit? Kasi your stomach has a gastric lining which is actually protected by mucus that, prevent your stomach from perforation when you are secreting a lot of acid. Now, in the case of intestine, they don't have what? Mucus protecting the intestinal lining. Thus, your body has to do something with the acidic time coming from the stomach before it goes to your intestine. Kasi pagdating niya sa intestine, tapos acidic pa rin siya, sunog lahat ng intestines mo. Thus, your body would be what? Stimulating your pancreas to secrete what? Bicarbonates. Bicarbonate together with the different enzyme, the bicarbonate would neutralize the acidic time present in the small intestine. Therefore, neutralizing it, increasing its pH up until what? pH 10. Because at pH 10, your intestinal enzymes would work best. What are those intestinal enzymes? Your pancreatic amylase, your disaccharidases, maltase, lactase, sucrase, they best work at pH 10. So far, naiintindihan ba yung lecture?
Yes po.
Okay. So, how do enzymes regulate your chemical reaction? So they do it by a reversible covalent modification or zymogen cleavage. They do it by allosteric regulation. They do it by reversible covalent modification. And they do it by genetic control. So when you say irreversible covalent modification or zymogen cleavage, as I mentioned a while ago, you have enzymes which are inactive in nature pa. And you call them what? Your zymogens or coenzymes. They need what? To be activated. Example of which would be your pepsinogen. The activator is hydrochloric acid. They need to undergo covalent modification or zymogen cleavage for them to become an active enzyme. So when pepsinogen is acted upon by your hydrochloric acid, there would be a cleavage of your pepsinogen, making pepsinogen now pepsin, which is in active form. Another enzyme would be your chymotrypsinogen. So chymotrypsinogen is also a protease, pancreatic protease. Now this is secreted by your pancreas. So it cleaves peptide bonds which are adjacent with your to arginine or to leucine. Okay? So they cleave that. Arginine, leucine, tyrosine, or asparagine. So chymotrypsinogen cannot act on your peptide bonds. It cannot catalyze your peptide bonds, not unless it becomes in its active form. So for it to become an active enzyme, it has to be acted upon by your trypsin. Trypsin initially is an enzyme also that is secreted by your pancreas. They're also non-pancreatic proteinases. Trypsinogen, would actually is an inactive enzyme that has to be activated by your enterokinase or trypsin itself, because trypsinogen is an enzyme that can undergo autocatalysis. So when you say autocatalysis, kaya niyang i-activate yung sarili niya pag na-activate na siya. So trypsinogen, yung activated form niya, trypsin, can become its activator. So, kaya nga, ang trypsinogen can undergo what? Autocatalysis. Now, trypsin now can activate other pancreatic proteases like your chymotrypsinogen. So, chymotrypsinogen is a zymogen. It has to be cleaved by your activator, which is your trypsin, to become its active enzyme, which is chymotrypsin. As you can see here, chymotrypsin can hydrolyze peptide bonds, which are adjacent to leucine, tyrosine, and your asparagine. So from there, you'll be able to separate these different amino acids. So again, this is what I've been mentioning a while ago. So you have your different zymogens. Zymogens are inactive enzymes, and they're usually your gastric and pancreatic proteases. Now, carbohydrates and lipids, they don't need what? They don't need activators, because by itself, the enzymes for catalyzing your carbohydrates and lipids are already active in nature. How come your pancreatic proteases or the proteins in catalyzing or hydrolyzing your protein are inactive in nature? Bakit kaya? Bakit kaya? Anyone from the class? So yan, hindi ko makikita yung chat group kapag mag-chat kayo. So anyone from the class would want to answer this? Bakit kaya ang enzymes nyo sa protein, inactive siya? Bakit sa ibang molecule naman like carbohydrates and lipids, hindi siya inactive? What can be the reason behind that? It's because most
They don't.
yes.
So they don't digest the pancreas itself.
Yeah, that's good, because most of your cells are actually made up of protein, right? If you would go back to your cell membrane, abundant or majority of your cell membrane is made up of what? Protein, di ba? Followed by lipid, followed by carbohydrate. So that's the reason behind why your pancreatic proteases are made as such, are made as zymogens. Otherwise, you'll be digesting your own pancreas. When this happened, okay, actually this can happen among patients who are what? Suffering from hemorrhagic pancreatitis. Have you ever heard of that kind of disease? Pancreatic hemorrhage or pancreatitis, acute hemorrhagic pancreatitis. So for example, nag-drinking spree yung mga magbabarkada, tapos nagpulutan. Then natulog kagad after nag-inuman, tapos isa sa kanila hindi nagising. Tapos nung dinala sa hospital, nung inotopsy, anong sabi ng doktor? Namatay daw dahil sa yes, pancreatitis, acute hemorrhagic pancreatitis. Okay? So sinasabi namatay daw sa bangungot, pero hindi yun bangungot ha, hemorrhagic pancreatitis yun. So another way would be allosteric regulation. So in allosteric regulation, okay, what happened here is that there's a regulation of activity, okay, of an enzyme caused by a reversible covalent non-covalent bindings of regulators at the site other than the active site. So allosteric enzyme can oxalate from active form to inactive form. So allosteric feedback inhibition, activation of enzyme in a biochemical pathway. So this is what happens. For example, you keep on sending positive feedback to this particular chemical reaction. When you produce a product from this chemical reaction, the product that you produce can send negative feedback, causing what? Later on, inhibitory feedback to your chemical reaction. So this can happen. Like for example, in digestion. So when you keep on hydrolyzing peptide bonds, okay, in your stomach, wherein you are actually secreting a lot of gastrin. Kasi gastrin is a hormone that stimulates what? Your acid secretion there. When you keep on producing a lot of product, because you are activating your pepsinogen, your pepsin, this product then that you produce would send negative feedback to your gastrin, inhibiting what? Your parietal cells to produce what? Hydrochloric acid. So, kung wala kang hydrochloric acid, walang mag-activate ng zymogens. So, this is basically your allosteric regulation. So, this is a diagram showing you that, for example, you have initial substrate, then you have intermediate metabolites, and then you produce your final product. So when you do that, the final product can send negative feedback, no? Rendering your chemical reaction what? It would stop later. So again, you can see here that once the substrate attached to your enzyme, you'll be producing products. These products, okay, if they are a lot already, they can actually bind to your allosteric site. Remember, allosteric site is the site other than your active site. So when a product binds to your allosteric site, you can no longer what? Produce what? You won't have your enzymatic reaction already because it inhibits the attachment of your enzyme to your substrate. See, you cannot attach your substrate not to your enzymes because of that. So it's not even fitted because it modified your site, okay, your active site. Okay? Kapag wala ulit na product, magmo-modify ulit yung active site mo para mag-bind yung substrate mo, then you produce products again. See? So final product accumulates in abundance, and because of that, the excess final product binds to the allosteric site on the first enzyme in the series of reaction. Okay, it inhibits the enzyme result. So when you inhibit the enzyme, you have produced less activity, less product. Then inhibition actually is reversible. So kapag wala ka ng products, ulit mag-aayos ulit yung active site mo as I've shown you a while ago. What about covalent modification? So covalent modification is modification of catalytic or other properties of protein by covalent attachment or of modifying a group, which is your phosphate. So enzyme, okay, enzymes can cycle between active and inactive. So they can have a slower or longer lasting effects. So this is actually, when we talk about covalent catalysis, we're talking about primarily breaking the bonds. Okay? So this involves transient bonding between an amino acid residue in the active site and that of the substrate. Generally, the active site of the enzyme should contain polar amino acid. What are your polar amino acids? These are your serine, threonine, cysteine, histidine, lysine, and arginine. So usually they make use of water, no? to break down the bonds. So, makikita mo siya sa mga may phosphate group. So the substrate should be polar, should also be polar, and the typical substrate are the phosphate groups. Not only your phosphate groups, they can actually have acetate group or they can have glycosyl groups. So any substrate that has a relative polar center, like an electrophilic center. So example of which would be your glycogen phosphorylase group. So they can be activated or inactivated. So glucagon can actually what? Can activate your glycogen phosphorylase while your insulin can inactivate it. So covalent modification is a reversible covalent modification of enzyme activity via phosphorylation and dephosphorylation. When it undergoes phosphorylation, it is catalyzed by protein kinase. When it undergoes dephosphorylation, you'll be needing a protein phosphatases. Again, as I mentioned, they can have immediate but slower or longer lasting effects. So here is an example of a chemical reaction, okay, that undergoes covalent modification. So as I mentioned a while ago, hindi lang phosphate group, acetate group din, saka glycosyl group din po pwede. Methyl group din po pwede. Okay? As you can see, they can undergo phosphorylation or dephosphorylation, adenylation or deadenylation, methylation or demethylation, and the rest. So what about genetic control? So genetic control is actually what? Your enzyme levels that can be controlled by controlling the expression of genes. Okay, so there are actually inducible genes that may be turned off and on. So this is actually the most sophisticated way of controlling enzymes. Kasi genes are DNA which encode for proteins. And proteins made through a process of transcription and translation of a particular gene. We discussed this in nucleic acid chemistry. Diba? So gene encodes proteins and enzymes are proteins. So some enzymes are said to be inducible by means the manufacturer of the enzyme can be turned on or turned off. If there is a lot of enzyme, the manufacturer can turn off the enzyme. Parang kanina sa allosteric, no? Feedback natin. But in this case, it is the gene now that turns off and on the enzyme. Kanina, you are sending negative feedback to turn off the enzyme. This time naman, you have the genetic control that turns off the enzyme. So the best example, okay, that is very popular, which is an inducible enzyme, is the enzyme lactase, which is encoded by your lac operon, or your LAC gene. So the LAC gene is the start of the group of clusters of genes known as lac operon, which can be turned on or turned off depending on the need of the enzyme lactase. The lac operon is usually found in what? In your E. coli or yeast. Okay? Yeast kasi they do not ordinarily metabolize lactose. In other words, they prefer glucose. Now, if you grow them in a media without lactose and have glucose, they will not produce enzyme lactase. Which makes sense. Kasi di naman kailangan yung lactase to digest lactose kasi nga puro glucose yun. However, if you start introducing lactose on a particular media, they would soon begin synthesizing an enzyme lactase by expressing lac operon. So how do they sense that there is a need for the enzyme lactase? How do they sense that lactase is not needed? This can be explained in the terms of lac operon. The lac operon is composed of LAC gene plus other genes. The LAC genes which manufacture the enzyme lactase is under the control of an operator or promoter. So, ito yung na-explain ko kanina, no? So, lac operon can be turned on or off. So, this is actually an example of your lac gene. You have your promoter and you have your operator. So, say you grow a yeast without a sugar lactose. So, in the absence of lactose, okay, there is a protein known as repressor. So, the repressor would bind to your operator. So, when it binds to the operator, the lac gene would what? Would shut down. Okay? Kasi walang makakabind sa promoter mo. The RNA polymerase that expresses the lac gene can bind to the promoter, but it cannot move forward because the road is blocked by the repressor. So what does it do? It would silence your lactase, just like in your yeast. Now say you have lactose, okay, in the particular media where you are growing now the yeast, okay, in the presence of lactose. So we now need the enzyme lactase. So what would happen there? So in this case, the repressor is there, and the sugar lactose is also there. So what does the sugar lactose would do? It would bind to your repressor. And then this changes the conformation of your repressor. Thus, what would happen? It can no longer bind to your operator. The laxing gene would now become unlocked. Okay? The repressor goes away, and then your RNA polymerase can bind to your promoter, and what? It can now express what? Your laxing gene. Kasi wala nang nag-block dun sa way kanina. So this can produce, there will be production of your mRNA that would be utilized for the production of enzyme lactase, which can now digest your lactose. So, parang sa med school din, you have your laxi gene, you have your operator, you have your promoter. So, if you have a lot of destruction, okay, your repressor is actually attaching to your operator. Your promoter is the one responsible for what? For encouraging you to study well, to pass. If you have a lot of destruction, you are creating repressor that attaches to your operator. So what would happen? Even if you have a lot of promoters, sinusuport ka ng parents mo, you have lots of books, we are giving you a lot of lecture, we're giving you points already, you cannot proceed any further. Because of the destruction, you are blocking your way to pass your subject. Hence, okay, passing or you are blocking what? Your dreams of becoming a medical doctor. So what will you do now? You go to an environment wherein you're not really that comfortable. Kasi when we are in an environment wherein we are not comfortable, like, you keep on studying, you block yourself from social media, okay? Hindi siya talaga convenient for us, di ba? And sometimes you are not talking a lot with your friends, you're not going out with your friends. Right now, kasi it's pandemic, talaga namang hindi kayo dapat na munang lumabas. Okay? So, you are in a media wherein you are creating what? Parang lactose, no? So, you are attaching your goals to the repressor, modifying the repressor, thus it cannot attach to your operator. And then, of course, when it happens, your RNA polymerase would attach to your promoter. Thus, if you keep on doing that, you keep on encouraging yourself, you keep on studying, you'll be able to pass the subject and you'll become a doctor talaga someday. So genetic control of enzyme activity, when you have lactose, it binds to the receptor protein. Receptor is inactivated. RNA polymerase can bind to the promoter region. Lac operon can transcribe into mRNA, then mRNA translated into lactase. Do you get it? Do you understand the lecture?
Yes po,
Yes
Doc.
po.
So let's go now to the different classification of enzymes. So in the book, you have different classification. Okay, so you have class 1, 2, 3, 4, 5, 6. So you have 1, which is oxidoreductase. So what does oxidoreductase do? Okay, it catalyzes it catalyzes the oxidation reduction reaction. Okay? Your transferase naman would catalyze the transfer of either carbon, nitrogen, phosphate containing groups. Okay? What about your hydrolases? It catalyzes cleavage bonds by addition of water. Lyases naman, it removes the carbon to carbon, carbon to sulfur, carbon to nitrogen bonds. Okay? What about your isomerases? It catalyzes the racemization of optical or geometric isomers. What about ligases? It catalyzes formation of bonds between carbon and oxygen, sulfur, nitrogen coupled to hydrolysis of high energy phosphatases. So remember their classification. No? So how are enzyme classified? So it's basically based on their action. So kung ano yung action ng enzyme, yun ang kanyang what? Yun ang kanyang action. Kung ano ang enzyme, yun ang kanyang action. Like for example, oxidoreductases. It would do oxidation, reduction, reaction. So like for example, like this one, no? You have what? What is this structure below here? Your CH3, CO2OH plus NAD. What is that? Anyone?
Ethanol.
No, that's not ethanol. That's your lactate. So you are catalyzing your lactate to become your pyruvate. Your lactate dehydrogenase. So actually, ano yan? Mali yan yung alcohol dehydrogenase. It's not alcohol dehydrogenase. That's your lactate dehydrogenase. You can find that in Liping Pot. Okay. So what about transferases? So what does it do? You are transferring your functional group. So in this case, what did you transfer?
Phosphate.
Yes. Kasi you have glucose on the left, then it would be acted upon by your hexokinase, okay, wherein you transfer what? Your phosphate, then you produce your glucose 6-phosphate. Okay, we have your hydrolases here. So in hydrolases, okay, what happens here? You have your phosphoanhydride. Okay, this would be acted upon by your hydrolases because you'll be removing what? Your water, no? So you add water to remove or to catalyze the CC bonds. Okay? And actually, you are removing water there. That's what happened in the hydrolysis. So now, when we discuss your digestion and absorption lecture, I want you to remember all the enzymes involved in the digestion, whatever it is, whether it digests your carbohydrate, protein, or fats. They are all hydrolases. Kaya lagi inyong maririnig dun sa lecture natin, hydrolysis. Yun lang yung chemical reaction doon. Kasi lahat sila hydrolysases. Another enzyme would be your lyases. So in lyases, it cleaves your carbon to carbon, carbon to oxygen, carbon to nitrogen, generating your carbon to carbon bond again. So just like your pyruvate, it will be acted upon by your pyruvate decarboxylase. You'll be removing the carbon dioxide there. Okay? You form your what? Your acetaldehyde. Isomerases. In isomerases, okay, you are actually what? Catalyzing isomerization. Parang ano lang siya, mirror lang siya. Diba? So, take a look at fumarate. Pag pinaggawa mo silang siopaw or parang nagpapalaman ka sa tinapay, no? Pinagdikit mo silang dalawa, they are actually what? Mirror, no? Mirror image. What about ligases? There is a condensation of two substrates splitting of ATP. So again, example of which would be your pyruvate that is acted upon this time by your pyruvate carboxylase. Kanina, do sa lyases mo. In lyases, it is acted upon by removing of what? Carbon dioxide. No? Kaya yung pyruvate mo, which is acted upon by pyruvate carboxylase, naging acetaldehyde siya. Dito, si ligases mo, it is acted upon by your pyruvate carboxylase. You add carbon dioxide to your pyruvate. Your pyruvate now becomes what? Oxaloacetate. Okay? So in summary, we discussed enzyme as your biologic catalyst. We discussed that with the help of enzyme, you are lowering the energy of activation. You are producing a spontaneous reaction, which doesn't necessarily mean you have a faster reaction rate, but you are creating a spontaneous reaction. Enzyme activity is affected by the following factors. pH. If you have an increased pH, you are denaturing or deactivating your enzyme. But take note, some enzymes works best at lower temperature, like pH 3 to pH 1. Some enzymes works best at pH 10, like your intestinal enzymes. For temperature, if you have decreased temperature, you are inactivating your enzyme, but you are not actually destroying your enzyme. As you return back to the normal temperature, it gradually recovers. But if you subject your enzyme to a very high temperature, you can denature, damage your enzyme. Substrate concentration. When you increase your substrate concentration, you are increasing the chemical reaction. But again, remember to a certain point. Because when you increase your maximum activity, no matter how many substrates you add in a concentration, you won't be able to what? To produce more substrate because you've increased your maximum concentration. And your enzyme is regulated by what? The following, as it can be stored by zymogen, which is your inactive enzyme. They have to be activated for them to undergo what? Chemical reaction or your catalysis. You can have your what? Allosteric regulation, wherein you are sending negative feedback or positive feedback. You can have covalent modification, no? Either in your phosphate group, in your acetyl, methyl, or glucosyl group. Enzyme induction through gene regulation, we discuss your lac operon. Okay? Your lac operon would have the different, your LAC-C gene. And remember, you have the different parts. You have your promoter, your operator. Your operator, that's where your repressor would attach to. Okay? When your repressor attached to your operator, there won't be your promoter can no longer proceed to your LAC-C gene. Therefore, your LAC-C gene won't be produced because there is a blockage in the catalytic pathway. But when you subject a certain microorganism like your yeast in the lactose, when you put lactose there, the lactose can act on your repressor, modifying your repressor. Thus, your repressor can no longer bind to your operator, and then your promoter then would proceed through its reaction. Thereby, you will be producing what? Relaxed CG. And then we discuss the different classification of enzymes. So the classification would be what? Your oxidoreductase, transferase, hydrolases, lyases, isomerases, and ligases. So we end with part 1. Do you have any questions? Do you have any questions? Okay, you can now chat in the chat box if you have any questions. Or I proceed to part 2. Wala? None? Yes, Christina.
Ma-clarify ko lang po, pag may nakabind na po sa allosteric site, tapos may another enzyme na gusto pong mag-bind sa active site, hindi po siya makakabind as long as may nakabind po dun sa allosteric site. Tama po.
Yes, that's correct. Kasi you're sending negative feedback already.
Hindi po ba?
Any more clarification? Paano po nagkaka-pancreatic failure? Bakit, Mark? May plano ka bang uminom mamaya?
Yun po kasi yung po kasi sa inyo,
No, not necessarily. Kasi may mga tao kasing genetic predisposition for the pancreatic failure. Kasi mapapansin mo naman, not all not all people who sleep after drinking spree would have hemorrhagic pancreatitis. So, ang pinakamagandang solusyon dyan para hindi ka magka-pancreatic failure, alam mo kung ano? Huwag ka nang uminom bago matulog. Minom ka maaga pa lang, mag-start yung klase, ganun, inom ka na para hindi ka makatulog kagad, di ba? Huwag na uminom. Okay. Would activity affect enzyme activity po? Like exercises in relation to lactase dehydrogenase? Yes, of course. It depends, di ba? Kasi when you're exercising, umpisa, di ba, hindi masakit yung katawan mo. Diba? So, kasi you are actually undergoing aerobic respiration. You have oxygen, and it's okay. You're producing what? You are not producing lactic acid. But there would come a point that you are actually shifting to anaerobic respiration because you are what? Maximizing already your enzymes, okay? Your oxygen as well. So when you shift to your anaerobic respiration, you are decreasing your pH, you are producing lactic acid already. So sumasakit na yung muscle mo, nagka-cramps ka na. So yun ang sasabihin sa parang yun ang way your body telling you to stop na what you're doing. Any question pa? Questions? If na-remove na yung naka-attach sa allosteric site, pwede na po bang mag-bind? Yes, pwede nang mag-bind. Okay. Balik tayo doon ha. I'll show you the illustration I made a while. Ayan. Nakikita niyo yung slide? Can you see the slide?
Yes po.
Ayan. As you can see, no? You have your allosteric feedback there. You have your allosteric site, your active site. You have your substrate. So when the substrate binds, okay, to your active site, okay, then you produce your what? Your products. When you produce your products, pag konti lang, okay lang. It won't bind to your allosteric site. Right? Pero, pag dumami yung products mo, yan na. Magbabind na doon siya sa allosteric site. When it binds to allosteric site, look at your active site. Nagkakaroon ng modification. Okay? So pag nagkaroon ng modification ng active site mo, ito na naman ulit, may magbabind ulit ng substrate, hindi siya makakabind kasi hindi siya swak. Right? So pag hindi siya nagbind, hindi siya swak, hindi ka magpuproduce ng products. Pero pagka nawala na yung products mo, syempre mauubos yung products mo, di ba? your active site again would go back to its normal form. Thus, your substrate now can bind to your active site. Now you can produce what? Your products. Do you get it? Let's proceed now to the part 2. So,
Yes, no.
okay, so let's proceed now to the part 2. So this is the part 2. We're going to discuss now the enzyme kinetics and inhibition. So for the part 2, we'll discuss the concepts of rates, reaction rates, and kinetics. We discuss the Michaelis-Menten curve, the Lineweaver-Burke graph, and enzyme inhibition. So at the end of this lecture, I want you to remember the following. You can discuss and you can explain the reaction orders. You can discuss and interpret Michaelis-Menten curve, Lineweaver-Burke graph. You can determine the Km and the Vmax and interpret their significance and meaning. You can explain the types of inhibition and determine and differentiate inhibition types based on the Km and Vmax. So ito yung pinakaayaw ng mga med students. So pag narinig nila si Michaelis-Menten, si Lineweaver-Burke graph, narinig nila si Km tsaka si Vmax, sobrang na-allergy sila dyan. So, who among you here took medicine because you thought medicine has no math? Sino? Sino sa inyo? Wala. Lahat kayo, feeling nyo may math yung medicine? Ang medicine actually maraming math. Ha? Okay? So, one of the math in medicine would be in enzyme. Hindi ko kasi makita yung shot eh. Yan. Sa NMAT po, may math kasi. Oo nga naman. So, kasi karamihan, nagme-medicine daw, wala daw kasing math sa medicine. Pero on contrary, maraming math. So, what is rate? So, rate is measure of change over period of time. So, for example, you travel 10 meters in 5 seconds, the rate is 2 meters per second. So, what is reaction rate? So, reaction rate is formation of products over period of time. Ang rate, it has the speed, it has the velocity, and it measures of change over a period of time. While your reaction rate, this is actually the formation of products over a period of time. So, by this one, you would want to know kung ilan yung product na mapoproduce mo over time. So what is the concentration of products after 10 seconds? You have 10 particles per ml per 10 seconds. So the reaction rate is 1 part per ml per second. So what's the relationship between substrate concentration, which is your R, and your reaction rate in uncatalyzed reaction? So you have this one, linear, and you have your, pataas lang siya. So what do you think is the answer here? Choice A or choice B? Actually, your reaction rate, relationship of your rate and substrate concentration is choice B. But again, remember, I keep on telling you kanina, when you reach the maximum capacity, even if you add a lot of substrate na, you would no longer proceed to have what? The chemical reaction. So, because of that, choice A is also an answer because this is actually pertains to your maximum reaction. Because when you reach the maximum capacity na, no longer how many substrate pa yung i-add mo, kahit 1,000 pa yan, yun pa rin ang reaction mo. Okay? So, choice A is actually your zero order. So, when you say zero order, even if we increase the reaction concentration, the reaction rate stays the same. It doesn't change. It is constant. The reaction rate is independent on the reactant concentration. If there are a lot of reactants, the reaction rate remains the same. Ang first order naman, if you increase the reactant concentration, there is a corresponding increase in the reaction rate. It is actually direct proportional relationship between the reactant concentration and reaction rate, and the reaction rate is dependent on the reactant concentration. The more reactants, the faster the reaction rate. So which of the two graphs accurately depicts as VO in an enzyme catalyzed reaction all throughout? So it is your first order. Or is it your zero order? Wala. Okay. This is your Michaelis Menten. So this is actually the one that depicts, okay, your enzyme catalyzed reaction. So it is neither flat, it is neither straight line or flat or ascending line. Anong tawag?
Hyperbolic.
Ha?
Hyperbolic curve.
It's your hyperbolic curve. This is your Michaelis Menten. So actually, your Michaelis Menten is a combination of your first order and your zero order. So when you are producing or when you are increasing your reactant concentration, your reaction rate increases. But there would come a time, even if you increase the reaction concentration, the reaction rate stays the same. So you have your mixed order. It's like this. So if you have a lot of substrate, and then you have your binding site here, sige, lagay ka pa, lagay. Produce what? Your product, lagay ka. Basta hindi pa napupuno, hindi pa saturated. Parang ship lang, hindi pa punuan. Diba? So, upo ka lang, tatawag lang yung nagtatawag ng sakay, pwede ka pang sumakay, pwede ka pang humabol. Pero, kapag napuno na, tatakbo na. So, kahit marami pang pasahero, hindi na ma-accommodate. Okay? See? Wala na. So, you get it?
Yes.
Okay. So, Michaelis Menten curve is that, we plot the reaction or substrate concentration against the reaction rate. So, this hyperbolic. So, in the first part where the substrate concentration is very low, this is most likely your first order. Kasi you increase the substrate concentration, there is a corresponding increase in the reaction rate. Hence, first order. However, when the substrate concentration is very high, it now assumes your zero order kinetics. So no matter how much substrate you add, it remains flat. It doesn't change. It remains constant. That's your zero order. So somewhere in between, there's a transition from your first order to your zero order. You call that what? Your mixed order. So why is it that when the substrate concentration is low, it follows your first order kinetics, whereas the substrate concentration is very high, it follows the zero order kinetics? This is due to the saturation of the enzyme. Remember, if you saturate your enzyme already, no matter how many substrate you add, you can no longer produce product. So when the substrate concentration is very low, there are more enzymes. Then there are more substrates. If we add more substrate, naturally more enzymes will be working. Therefore, there's a corresponding increase in the reaction rate. The reaction rate would concurrently increase as well with the subsequent addition of substrate. But if you keep on adding what? your substrate, there will come a point that all enzymes are already working. We have reached the point wherein no matter how much substrate we add, there's no change in the reaction because the enzyme is said to be saturated. So the reaction rate has reached is what we know as Vmax. So this is the maximum velocity of reaction. So no matter how much substrate you add, the Vmax won't change. Maliwanag yun, ha? So ito yun, yung kanina, yung paliwanag natin. So first order, then you have your mixed order there, then it would reach the point where zero order kinetics. Okay? So at low substrate, it obeys first order kinetics, more enzymes than substrate. Addition of more substrate increases reaction rate. All substrates are being converted to products. At high substrate, it obeys the zero order kinetics. All active sites are occupied, enzyme is saturated, maximum reaction velocity. Further addition of substrate cannot increase rate beyond Vmax. That's your Michaelis-Menten curve. Maliwanag, ha? So, hindi nga nakakatakutan si Michaelis-Menten curve. Now, pag na-reach mo na yung zero order kinetics, yun yung Vmax mo. Okay? Pagka naman nandun ka pa sa first order, ito yung Vmax 1 half. So, halfway through the Vmax is called Vmax 1 half. This corresponds to a substrate concentration. The substrate concentration and half of the Vmax is what you know as the Km. The Km is what? Your Michaelis constant. This tells us something about the enzyme's affinity to the substrate. So in this particular example, okay, kanina, if you are looking at your graph, okay, so there are two curves, the same Vmax but different Km. Okay. See? So you have Vmax pareho, pero you have different Km. So one has lower Km, the other one has higher Km. Between the two, which one do you think would have a higher affinity for the substrate? Which one has the lower affinity for the substrate? Now, when the Km is low, the Vmax is readily reached, even in low substrate concentration, because the enzyme is saturated easily. So, parang ano yan, ito yung mga mainitin yung ulo. Konting kibot mo lang, konting mali mo lang, mainit kagad yung ulo. Yan yung low Km. Nare-reach mo kagad yung Vmax nila. So, nasa-saturate mo kagad sila, napupuno mo kagad sila. Now, yung Km2 naman, wherein you have higher Km, sila yung mahaba yung pasensya. So, matagal-tagal mo rin pinainit yung ulo nila bago nila na-reach yung Vmax. So again, low Km means high affinity for substrate, okay? And the reverse would be true. So when the Km is very high, it takes a lot of substrate for the reaction to reach Vmax, which means the enzyme is not easily saturated. High Km means low substrate affinity. We always take a look at the Km to determine the enzyme's affinity to substrate. Gets nyo, ha? So, the Michaelis-Menten can be described using the following formula. So, VO is your reaction velocity, which is equals to Vmax multiplied by substrate concentration. So, kanina, madali lang. First order, zero order tayo. Ang dali intindihin. Tapos, inintroduce natin si Michaelis-Menten. Siya yung hyperbolic curve na merong first order, tapos magiging zero order. Ngayon, pahihirapan na natin lalo. Kasi syempre, medical students na kayo. Papahirapan na natin kasi papasukan natin ng formula. No? The VO is the reaction velocity which is equals to your Vmax multiplied by the substrate over Km plus, which is your substrate then. So it is this is non-linear relationship. So the formula describes the relationship between velocity of enzyme catalyzed reaction and substrate concentration. So how do we change Michaelis-Menten to line-Weaver-Burke equation? So we try to convert the Michaelis-Menten curve into linear formula so that we can readily derive Tmax and Tm. So you get the reciprocal of your Michaelis-Menten, separate the Km and substrate, then cancel and simplify. So do you recognize the formula? It is your y equals mx plus b. That's your Michaelis-Menten, kaya nga linear siya. Okay, you obtain the reciprocal, you separate the Km, the substrate, cancel, simplify. So, this is your what? Your y equals mx plus b. So, pagka ganyan naman yung ano mo talaga, linear talaga yung lalabas na graph mo. Tama? So, when you plot, okay, so this is linear plot already. So, what you plot now is 1 over substrate versus 1 over your VO. Okay, so your 1 over VO is the y and the x is 1 over substrate. 1 over V max is the y-intercept, and if we extend all the way to the negative side, the x-intercept is the negative of the Km. Yan na. So, mat na. Ayaw nyo na. Pero kailangan kasi sa exam tayo. So, let's try to have an exercise. For example, you want to determine the V max and the Km. So, this is the data given. Okay. So, we will try to plot this data S versus your Michaelis-Menten. So for us to say that this is Vmax, there should be flattening. If we continue adding more substrate, there should not change. We should not change anymore. But if we keep on adding more substrate and it keeps on rising, the substrate, it means to say it has not yet reached the Vmax. So we have to convert this to line Weaver-Burke. So, which is the reciprocal of your Michaelis-Menten. So, in x, it would become 1 over s, and in y, it would be 1 over t0. So, you have to plot it. Okay? So, ito yung Michaelis-Menten mo. So, kapag binigyan ko kayo ng formula na ganito, huwag kayong nga, ha? So, anong gagawin nyo? Kailangan nyo siyang i-convert into your line, Weaver-Burke graph. No? Pagka i-convert nyo yan, kailangan yung reciprocal. Reciprocal, meaning to say, yung sa x side mo, dapat 1 over s, so y mo magiging 1 over v. Kaya ang mangyayari, dapat linear, no? Kailangan reciprocal. Kaya ganyan ang mangyayari kapag ka nag-reciprocal ka, no? 1 over s, 1 over v. See? See the difference? Huwag niyong kakalimutan mag-reciprocal, ha? So extrapolating this all the way to the negative side, we can now derive the Vmax from 1 over Vmax, okay, and also derive Km from 1 over Km. See? Itigil yan. That's your Vmax. Kailan nag-intersect. Diba? So recap. We discussed the Michaelis-Menten. It's a hyperbolic curve. Because dahil hyperbolic siya, walang katapusan, you'll be reaching your what? Your zero order kinetics. Now, in this one, we want to know when you saturate your enzymes already, when you stop having affinity of your substrate to your enzyme. We want to know it by doing what? Your Line-Weaver-Burke graph. Okay? In Line-Weaver-Burke graph, you have to make a reciprocal to your Michaelis-Menten. Okay? Now, let's go to the different enzyme inhibition. So enzyme inhibition will be as follows. You have the irreversible and you have the reversible. Under irreversible, you have your competitive, non-competitive, same as your reversible. So let's discuss them first one by one. Kasi ang irreversible mo, konti lang, no? Like when we discuss cyanide poisoning, you'll find out later on that it can be both reversible and competitive and non-competitive. But it's a reversible. Viagra also can be a competitive and penicillin. So in reversible inhibition, okay, so this is your competitive, no? So we studied the Vmax, Km, Lineweaver-Burke, and Michaelis-Menten because we can use this to determine the different types of enzyme inhibition. So in competitive inhibition, the substrate and the inhibitor compete for the same binding site, which is the active site. See? You see the substrate, you see the I, that's the inhibitor. It's the substrate that binds to your enzyme, well, that's good. If it's the inhibitor that binds to your enzyme, the enzyme would turn inactive. This is the reversible type of inhibition. The inhibitors often resemble the substrate structurally. They are said to be molecular analogs. So how can we make sure that the substrate is the one that will bind to the active site? You increase what? You increase your substrate. Diba? Para walang chance si inhibitor na mag-bind sa active site. Okay? So if you keep on adding or if you have more substrate, it means that there is a greater likelihood that it's the substrate that will bind to the active site rather than the inhibitor. We can still reach the Vmax as long as we have lots of substrate. The Vmax doesn't change. We have two curves here. No? So we have here the blue one, which is without the inhibitor, and then the red one, which has the inhibitor. So as you can see in the red one, no, in the presence of competitive inhibitor, the substrate can compete against the inhibitor. So notice the Km has been reset to a higher value. So in competitive inhibition, the Km increases, but the Vmax doesn't change. So how does it look like in your Lineweaver test? Ano yan? So in reversible competitive inhibition, okay, you increase your substrate para walang chance si inhibitor mag-bind. Okay? Hindi ka maagawan. Okay? So yun yun. So para makamit mo yung isang bagay, okay, so kailangan marami ka talagang i-offer. Like for example, if you want to pass the subject, you have to offer not only, you are not supposed to be very good in reporting only, you should be good in your shiftings, you should be good in your major exam. So you increase your chance of binding to your active site wherein the product would be what? Passing the subject. Pero, kung magaling ka lang sa report, tapos, ang dami mong problem sa shiftings, baksak ka parate, even your major prelims baksak, you are increasing the likelihood of your inhibitor. Therefore, walang what? Walang product na mapuproduce. So, hindi ka, what? Hindi ka pa rin papasa later on. That would be the product. So, what about non-competitive inhibition? So, they're not competing with the same binding site. Ang competitive inhibition, they're competing with the active site. Pareho lang. Patas ang laban. No? So, parang yung lalaki na dalawang lalaki may gusto sa isang babae. So, isa lang ang pinupuntirya nila. Okay? Isa lang yung talagang target nila. So, yun lang ang active site nila. Okay? Dito naman sa non-competitive inhibition, they are not competing with the same binding site. The substrate binds to the active site. The inhibitor binds to the another site, which is called the allosteric site. So the inhibitor can bind to the free enzyme. It can also bind to the enzyme that is already bound to the substrate. Inhibitors never bind to the active site. So you have both the enzyme inhibition, enzyme inhibitory substrate complexes, which are enzymatically inactive. So parang ganun. So, ito yung parang kampante ka dun sa jowa mo, tapos may best friend ka. So, akala mo si best friend mo, hindi naman agawin si jowa mo. Tapos, biglang nung umalis ka, sa allosteric site pala nagbabind si jowa mo, ay si best friend mo, so tinitraitor ka nila. So, akala mo all the while, okay kayo ni jowa kasi nakakabind ka sa active site, pero si best friend pala dun sa allosteric nagbabind. Kaya pala hindi nag-level up yung relationship mo ng jowa mo. Kasi merong nagbabind sa allosteric site. Okay? So, inhibitor can bind to the enzyme. So, na-mention ko yan kanina. So, can increasing substrate overcome inhibitor? Parang kanina, no? Ang sabi ko, kung kunwari, in-increase mo yung pag-aaral mo, in-increase mo yung shiftings mo, ginalingan mo sa SGD mo, ginalingan mo sa major exam mo, makakapasa ka, di ba? Kasi you're increasing your substrate. But do you think in non-competitive inhibition can do the same? Hindi. Kasi nga, sa allosteric site siya nagbabind. Eh, ang substrate mo, nagbabind siya sa active site. Right? It's the same thing. So, kahit ikaw, nag-e-effort ka sa relationship mo ng jowa mo, pero kung may best friend kang ahas na laging nakabind sa allosteric site, hindi talaga mag-propose for yung relationship mo. So, anong kailangan mong gawin? Tanggalin mo yung allosteric site at tanggalin mo yung inhibitor. So, there's no use. Kahit magluto ka pa ng masasarap na pagkain, kahit magbigay ka pa ng magagandang gifts, kahit lahat ng qualities mo na magaganda ipakita mo, it wouldn't matter because the inhibitor is binding to the allosteric site. Right? So what happens here? So even if you add a lot of substrate, it cannot overcome the inhibitor. Kasi nga, sa allosteric site siya nagpabind. The original Vmax cannot be achieved anymore even if you add a lot of substrate. It has decreased. The Km doesn't change. It remains the same. So in non-competitive inhibition, Vmax is decreased, Km is unchanged. See? This one. So this is non-competitive inhibition. Feeling mo okay na okay kayo, pero yun pala, yung nagpabind na inhibitor. Okay? So if you look at the line Weaver Burt plot, you would appreciate here that in your competitive, okay, the green line would actually, it is without the inhibitor, no? So the red line is the one with inhibitor. So notice that they have the same y-intercept, which means they have the same Vmax, which ultimately means that the Vmax has not changed. But in non-competitive, what do you notice? The green one is the one without inhibitor, the red is with the inhibitor. Notice that they have the same x-intercept, which means they have the same Km. Ultimately means that the Km is the same in the presence of inhibitor. Gets nyo? So this is the summary of what I've discussed a while ago. In competitive, the binding site is the active site. In the non-competitive, tandaan nyo, traitor yan. Okay? So, nagbabind siya doon sa hindi mo alam, sa allosteric site. So in summary, we discussed the two types of inhibition, which includes your competitive and non-competitive inhibitor. Again, in competitive inhibitor, you have the same Vmax, 1 over Vmax. So meaning to say, you just add substrate, you'll overcome your inhibition. In non-competitive inhibitor, you have the same Km. So no matter how many substrate you add, it would remain the same. Okay? So these are my references. So I make use of the Lippincott, Harper's, and Lehringer's. So that ends my lecture. Do you have any questions?